I am an anthropologist, and I do research on food security and nutrition, but I'm not speaking tonight. I will be speaking on May 24th, however. And I get this opportunity because of a special collaboration between Town Hall, the UW Engage program, and this year we've had the pleasure of partnering with Ada's Technical Books. Together, they host the series called the UW Science Now as part of the Seattle Sciences Lectures. Sponsors include Microsoft, Boeing, and the Bill and Melinda Gates Foundation. Each speaker's program tonight will run for about 25 minutes, including 5 minutes for your questions. Between each of the 3 speakers, we'll have a little bit of time for you to use one of the 2 gender-neutral bathrooms to my left, or get a drink, and then the next speaker will go. So, before I introduce our first speaker, I'd like to mention the next event in our science series. The Night of Talks in the UW Science Now series will be right here again at Ada's Technical Books on April 26th, starting at 7:30. Rachel will talk about vaccine delivery. Maria will talk about quantum computers. And Brita will talk about ice compaction in Antarctica. So be sure and get your tickets for that event as well. Okay, so to our first guest. There are many viruses that have little to no treatment or cure. Before successful therapeutics can be made, these viruses must be well understood. Our first speaker will tell us how she uses microscopes to visualize viruses at the nanoscopic level. Please give a warm welcome to Lexi Walls.
Good start. Okay. Hi, everyone. Thank you so much for coming out, and I'm really excited tonight to talk to you all about viruses. So the question that I want to start out with is, what does a virus look like anyways? And first, I want to ask a question. Who, when I say the word virus, thinks of something that looks like this? Nice, awesome. That is not what I'm going to be talking about tonight. Instead, I'm going to be talking about things that look more like this. Now, this is kind of a futuristic, kind of sci-fi representation of viruses, but tonight, hopefully by the end of the talk, I'm going to convince you guys that understanding what these truly look like at the microscopic level is really important for us and for the future of vaccine design. Now, this isn't the only place that we see viruses. We also see them in instances like this. Who has had the fortunate or unfortunate pleasure of having a virus that causes these feelings this year? Yeah, I'm sorry. And sometimes, if you can remember back to a long, long time ago, hopefully, who has had a virus that looks something like this? Yeah, okay. So viruses are everywhere, and not just in how we're feeling, but also in our pop culture. So it's in books, in movies, and not just in books and movies, but also in our news and magazines. So back in 2016, during the Olympics, Zika virus was a huge news story, and even this nice cartoon about the Olympic flame running away from a mosquito. Now, possibly my favorite representation of viruses in pop culture is the fact that we can give kids and our friends nice plush toys of viruses. So they really are everywhere. But all of this aside, the first question is, what is a virus? So when I think of a virus, I am a structural biologist, and so I think of what does the virus look like and how is it formed? And so the way that I think of viruses is I think of them like a letter. So letters, the first thing they have is an envelope, something to really hold in everything that is contained inside of that letter. They have a message, something important, something that you want to share with someone. And then the way that you share this with people is by having an address. And this address goes on the outside of the envelope. And this address really allows this message to get to its proper location. And finally, these envelopes have a way to basically protect their information. So this might play, yes. So this is a GIF of Sandra Bullock opening or attempting to open an envelope at the Oscars. And so this is kind of something that viruses also like to do. They like to protect their message that's inside. So how this relates to a virus, so viruses have an envelope, or basically a coating that holds in all of the important things. And this coating here is represented in blue. Viruses also have a message, and that message is the genetic material, or how viruses pass on their information from one generation to the next, how they propagate inside of us and eventually cause infection. And then finally, viruses have an address. And this address is in the form of protrusions that are on the exterior of the virus. So these are kind of how I like to think of the structure of a virus and what a virus really looks like. Now, viruses are beautiful. They can look all different sorts of ways. They can be spherical, icosahedral. You can see here there are protrusions coming out. They can be filamentous, long tubular protrusions. And then they can also be futuristic and look like nothing else we've ever seen before. And what's really interesting is that all of these viruses all have one goal: to infect and to propagate and move on. And so all of these things, while looking wildly different, have really a lot in common. Now, in order for a virus to infect, it needs to kind of accomplish a couple of things. So in this image, we have a virus, spherical, on the top of the image, and we have a cell. You can think of a cell as our host cell, our human cells, host cells. And for the virus to infect our cells, it needs to do a handshake. It needs to basically connect to our host cells in a specific manner. And this handshake is really how the virus first attaches and first sits down onto our cells. After the handshake is done, the door can be opened, and a virus is now allowed to go into the cell. And unfortunately, yes, there we go. The virus is allowed to go into the cell, and unfortunately, this means that infection is going to be successful at this point. So if we want to prevent viral infection, which can we all agree we want to do? Yes. If we want to prevent viral infection, we need to stop it before it gets to this point. And so David Goodsell is an artist who actually does a lot of biological drawings. And so I just wanted to share this really beautiful image. So what I showed before was very simplified and very clean, but these processes are not occurring in a vacuum. There's a lot of other actions and mechanisms that are occurring at the same time. And so for the virus to really touch down and make this handshake is in some ways a beautiful thing if you don't think about all the bad consequences of viral infection. Okay, so now I've told you how viruses can infect. And so now I want to talk a little bit about how we can work to prevent viral infection. So one of the first ways that we can prevent viral infection is through public health measures. So one, washing your hands, covering your sneeze when it happens, covering your cough. That was perfect timing. And if you are sick, staying away from people so that you don't share your viral infection with others. However, if these methods don't work, then we have a second line of defense, and that's the immune system. So once the virus gets into us, our bodies have a number of measures and ways that they can prevent infection from happening. And so these were the measures that were in place in the 1900s. And so I'm going to show an infographic where the number of infections of viruses in the US are depicted by spheres. So the larger the sphere, the more people that were infected by this virus. So what I hope you all can appreciate is that measles in yellow, the largest sphere, had a large burden on the US population, as well as many other familiar viruses that you're seeing. Mumps, the flu, smallpox, polio. And so this was really a challenge and a difficult public health problem. And so something major that has changed since the 1900s is the invention of vaccines. And so the way that I like to think about vaccines is like training wheels for the body. The vaccine acts as sort of a blueprint of the viral infection. And so you can imagine that if you receive training wheels before being thrown onto a bike, you're going to be more successful at riding that bike. And that's exactly how vaccination works. There are two major types of vaccination, but essentially it is showing your body a way that this virus is going to look to it in the future, allowing it to prime itself, prepare itself for eventual infection. And now, what's really amazing is that vaccines have been super, super successful. Look at the size of the spheres on your right. They are massive in comparison to what they are in 2010. Smallpox has been completely eradicated from the face of the earth because of vaccination, and polio is extremely close to being eradicated. Other diseases, you can see measles, that sphere went from massive to tiny. It's really amazing what vaccination has been able to do. And what I think is even better, a better way to describe this, is that in the 1900s, this is what biking looked like. And now, this is what we can do with all of these viral infections. And now, I think what you're all thinking is, we're done. We can go home. We've solved viral infections, right? But we know that that is not true. And so, unfortunately, if we take a look at the world and we take a look at the different diseases that have been reported, this is from 2016, what you can see is that there are still a lot of viruses that we have no cure for. Not even a vaccine, no antiviral, nothing. There's a lot of names up there that you might recognize: dengue, Zika, MERS. A lot of these we have no way of treating. And so that means that our work here is not done. And so we, as researchers and scientists, need to propose a new way to be able to understand and to be able to vaccinate better and design vaccines in a much better way. So right now, unfortunately, the landscape looks like this. The viruses are winning the race. However, I hope that through some of this work, we can really reverse this cycle and really switch who's winning in the race against virus or humans. So the question then becomes, how can we make vaccines more effective? And the way that I and a lot of my colleagues propose to do this is pretty simple, I think. We're going to look at them. We are going to take a look at these viruses and see if we can understand how they are working in order to prevent them from working, is the eventual goal. Now, I want everyone to take a step back, pretend you're not at Ada's anymore, and pretend you are in IKEA. You're walking in, you planned a 20-minute trip, and 2 hours later, you leave IKEA and you have a bunch of boxes of furniture. And now, imagine you're going home and you're about to assemble all of this furniture, but there's no map, there's no directions, there is no information for you to assemble this. The picture of you is going to look something like this. And this is really what we as researchers look like with viruses and vaccines now. If we don't know what these viruses look like, how can we assemble an effective way to prevent them from working? And so the goal is to get an IKEA blueprint of these viruses in order to make a better vaccine. So how are we going to do that? We're going to use microscopes. And so this is kind of a typical image, not the one of them, only one of them is a typical image. This is a typical image of a microscope. However, this is not the type of microscope that I use. The type of microscope that I use is called an electron microscope. It is 16 feet tall, it costs $10 million, and it weighs multiple tons. For scale, that's me, next to our beautiful microscope. And if you think that this is complicated and huge and large, just look at the inside of it. It is crazy the number of connections and the number of things that have to go right for this microscope to work. But using this microscope, we can do incredible things. This microscope can look at a scale unlike any other microscope before. This microscope can look at the scale of cells all the way down to individual atoms. And the things that I'm going to focus on in the rest of the talk are highlighted here: cells, viruses, and proteins that are on those viruses and cells. So to give you an idea of the power of this microscope, this is a typical textbook image of a cell. It looks beautiful. There's a lot of detail, and it's quite impressive what is really in our cells. However, when you look at this cell with an electron microscope, this is what it looks like. We can get incredible detail with this work. We can change what the textbooks are showing us. That is powerful. That is amazing. It is so exciting to be working on these techniques and on these microscopes and instruments. Now, how does this work? How do we get these images? Okay. So how do we get these to work? So we have whatever we want to image, a cell, a virus, a protein, whatever it is. We have that, and we shoot it with electrons. When we shoot the sample with electrons, we get a 2D projection, just like an image, which you can see below. Once we have this image, typical image looks something like this, we can computationally take this image and say, these are the parts that I'm interested in this image, highlighted here in yellow. We can extract them, align them, and understand at higher and higher detail what they look like. We can do this for various views of the same image, of the same particle. And what this allows us to do is construct a three-dimensional image and three-dimensional model of what we're looking at. This allows us to get to really, really fine detail, allowing us to really understand at the molecular level how these various things that we're looking at look like. This technique is so powerful that it actually won the Nobel Prize in this past year. And so what we hope to do is to apply this technique to viruses and vaccine research. So how do I plan to do this? Well, to start, I'm going to take this virus and I have to think, okay, if I were going to design a vaccine, what would be the most intelligent way to do this? What would be the best way to do this? And the way that myself and a lot of people in the field have determined to do this is to really go after that address. So the protrusions on the outside of the virus. And the reason we think this is if you sent a letter with no address, it wouldn't go where it was planning to go, and the message wouldn't get where it needed to get. We would prevent viral infection. And so the way that we want to do this is to basically prevent this handshake from happening. So some way that I kind of imagine this when I'm working on it is imagine a bear climbing a tree, but instead of being able to climb a tree, you prevent it from being able to use its arms. For example, you put boxing gloves on them, or you chop them off, whatever you want to think of. But essentially, you're removing the bear's ability or the virus's ability to latch on and to enter whatever it is it's trying to do. And so this has been done for many viruses, and we wanted to show that we can do it in the exact same way, but for a variety of different viruses. So again, I'm showing David Goodsell's really beautiful artwork. So this is a virus, and the protrusion that you're seeing colored in yellow and red. And typically, the virus needs to attach to the cell in some way. And the cell and its attachment is shown in blue. So the goal is, can we find some way to block that interaction, to prevent the bear from using its arms? And so the virus that I'm interested in are called coronaviruses. Has anyone heard of coronaviruses? A couple, that's great, awesome. Coronaviruses are called this way because they have a crown of these protrusions, these spikes that are extending from the viral surface, which you can see here in this 2D projection and also in the 3D image. So coronaviruses, if I was giving this talk in 2002, it would look something like this. I'd say, oh, coronaviruses, they're so bad. They can infect pigs and cows and cats. We really should care about these. It's terrible. It's bad for livestock, bad for our pets. However, that image really changed in 2002 with SARS, or severe acute respiratory syndrome. So SARS was first discovered in 2002, and actually 10,000 people got infected, and there was about a 15% fatality rate. There's no cure, there's no treatment, people just had to wait it out. And we still don't have any cure or treatment for this virus. This happened again in 2012 with Middle East Respiratory Syndrome, or MERS. And unfortunately, this infection is still ongoing. And again, no treatment, no cure, no way of helping people who get infected. And what's even scarier is that a couple months ago in SeaTac Airport, I actually took this image. If you go to SeaTac, you will see signs of warnings about Middle East Respiratory Syndrome plastered all over the airport. So this is a real and true issue that we need to be dealing with. And so the real question is, have I been successful, right? That's what everyone wants to know. So what I've been able to do by combining coronavirus research and cryo-electron microscopy is I was able to solve the first structure of these protrusions at atomic resolution detail. And so you can see them protruding here in purple. when I was able to do this, we've done it now for multiple different coronaviruses, human infecting and animal infecting, and we've learned a lot. And so we wanted to say, okay, now we know what the virus looks like. Now we know what the address of this virus looks like. Can we prevent the address from being functional? Can we prevent the cell attachment? And so, again, just to remind you, this is what those protrusions look like. This is how they want to attach to the cell, and this is how our goal is to block the infection. And so, taking the map of the coronavirus spike, typically it will try and attach to the cell at this location. And then what I've been able to show is that we can actually get molecules, things to bind and to block this interaction. And so where it would typically be interacting with the cell, it is no longer able to do so. We've successfully been able to prevent infection and understand this at the microscopic level. We've been able to do it multiple times with multiple different spikes, with multiple different coronaviruses. And so with this is kind of the first step to being able to design an effective vaccine against coronaviruses. Ooh, sorry. And so my goal and the goal of the field is that eventually this race will be reversed by using these powerful microscopes, these powerful techniques, and really understanding how viruses work at the microscopic level. So with that, I have a lot of people to thank. I did not do any of this work alone. I work in a wonderful lab. We're the Wiesler Lab in the Department of Biochemistry. I also have a large number of collaborators that are international and here in the US. I was funded by the NIH through a training grant. And I would also like to thank ADAS, Town Hall, Engage, and really all of you for coming and giving me your attention for the last couple of minutes. So thank you, and I'd love to take any of your questions. I think they are some of the most creative things that actually live on this planet. They can infect from bacteria all the way up to humans, and they do so with many different ways, shapes, and pretty much everything. They are so diverse and so incredible.
I'm curious of the timeline between your research and actually getting a vaccine that can be used
Yeah.
for the general population. What's that timeline and what's the next big hurdle?
Yeah, so if we could predict it, I would have $1 million. So this is really still in the basic side of research. So the goal is to show that this is feasible and functional, and then ideally we would be able to move forward into clinical trials. So what I've shown here is that we're able to block infection, but we also have to make the body be able to block infection. So there's still a number of hurdles that we have to overcome, but I mean, in theory, in 20 years, maybe not for coronaviruses, but for other viruses where we're doing this work, we could feasibly have something in the pipeline. Yeah?
So this technique you were talking about, electron microscopy, seems like it would be really good for things that are static, things that are not moving around. Do any viruses have like wiggly addresses, and how do you deal with that sort of thing?
Yeah, so the question is, so when we're doing the imaging, how do we deal with things that are moving around? So something I didn't address is that when we're imaging, we've actually frozen these things at minus 180 degrees Celsius. So they are completely frozen in time. So even if they were moving in real life, we've actually frozen them. And the goal is to be able to average these all together like I showed in the slides. If things are moving, it's more difficult to average, but there are a lot of computational ways we can get around that, and we're getting better and better every time. But yeah, that's a good point. Yeah?
Are you confident yet that the attachment mechanisms of the viruses you're studying, all in the one family, are those mechanisms the same throughout that family of viruses?
Yes. So the short answer is no. They all use different attachment factors, and we actually think that they are recognizing in many different ways, and that's kind of one of the reasons these viruses are so intriguing, is that they are really diverse. So even though There are multiple that infect humans. They're using different ways, different handshakes, totally different types. But if we can still block, we don't care specifically how they're interacting with the cell as long as we can majority block that region. But yeah, that's a fantastic question.
So along
Yeah.
those lines, I mean, viruses mutate.
Yes.
Right? So does this virus mutate as much as a cold virus?
Yes, so all viruses kind of have different mutation rates. Luckily, I guess, for these ones, they're not super fast within a specific virus. So within SARS, it's not mutating too quickly. But yeah, it's a good question and good point. Yeah?
How long does it take to get one of those like cell images? How long does it take?
So the cell images, if you have everything working perfectly, perfect sample, one of those images of the cell will take about 2 hours. But the images more of the single particles, those each image takes about 2 minutes, but you need about 1,000 to 2,000 of them. So it does take a while, but yeah. Yeah. Yeah, so the question was, where do you get your samples from? So most of the time we grow them in the lab. So totally recombinant, no human, no other thing is involved. We completely purify them from whatever source we are choosing to use. But no one needed to be infected for any of this research to happen. All right, if there are no more questions, thanks so much.